Great, thanks very much. So if people will line up at the microphone for questions, and Marty, you can kick us off.
Thank you. Janet, I just wanted to respond to a few of the things that you said. You first said that you like the iSpy protocol, which I like very much as well. And to simply say that what I have in mind in my proposal is to take iSpy to the next level and be able to apply it to all of the patients who are not engaged in formal trials, to essentially set up what Andy Grove has talked about when he talks about e-trials, to have a knowledge about each patient de-identified, such that one can group those patients in whatever cohort one needs in order to be able to analyze the data of what you have, the treatment arm you're looking at, against an appropriately stratified control group. Secondly, you talked about Mini-Sentinel as an infrastructure that you'd like to leverage. And I'm all for leveraging infrastructure to the extent that we can take something that's built. I don't want to reinvent anything. I would love.
Everybody else does.
Well, no, I think that the only way I'm looking actively for anyone who has infrastructure, and I heard many good projects that could be built on, but those have to be opened up for the driving purpose of being able to inform patient treatment decisions. You talked about, trying to read my own writing. Where's this? You talked about journals, and I guess you did, about journals. And one of the things about journals is they work very well for science, but they're very slow for medicine. The idea of being able to take a year of a review cycle, and not to mention escrow, when there's something in that paper that might affect the patient's life, is, to me, speaking with my survivor hat, unacceptable. And so there's an opportunity to be able to take what we're talking about in a rapid learning community and be able to make that an open peer review, post-publication peer review type of process where it gets widely scrutinized and but available to patients and doctors immediately with the appropriate strength of evidence indicated.
Thanks very much.
Excuse me, one last comment. You talked about standards being very important. And this is something I know a lot about because I developed many successful businesses in the internet that became successful precisely because they exploited the lack of standards in an area which really demanded interoperability. And so it's important to remember that interoperability is the goal. Standards are a means to that end. And when you have problems like one talks about females and one talks about 1-0s, that it involves a relatively small amount of time, typically a day or two, to be able to take a file in whatever form and map the metadata for that file once to whatever standard model you want to use, and then that data is interoperable with all the other data that has taken a day or two to map it. It's very easy for computers to translate females into one.
I know, and that's a bad example, all right? So I use it because it's funny, and we do that. I mean, that is done already, and there's, say, a common data model. It's not easy if the doctors don't enter the phenotype of the patient, and that is, complexity of biology is not like the complexity of like marketing, in my opinion. And that's the problem we have. We have to have some this is where the data people and the biology people or the medicine people like go on different tracks, because we want to reduce sources of variability so we can make sense of what is an incredibly complex situation. And the data people say, oh, we just have enough data and we'll figure it all out. And it isn't true, okay?
That's
not true.
That's not what I said, though.
I know, but, well, that's what the sort of the IT side says. If we have enough data, well, we can map it or whatever. But what we've seen is, you know, you have to have the patient's phenotype in there somewhere. You have to understand what the patient has, and if you can't construct that from bridging or a model or something like that, it has to go in there. So you have to constrain that to get entered somehow. That's what the data standards are really for. Yes.
Having been around Dr. Woodcock and Dr. Temple for 40 years, I know the rigor and quality issue. My biggest concern, and we were talking about journals, and the biggest problem we have is the publish or perish. If you don't have a resume with 50 articles, you may not get tenure. And we don't emphasize quality, we emphasize quantity in that process. And I think that we've all been exposed to it today in questioning quality of the data. Everyone says don't put Part 11 in as a requirement for a lot of stuff going on. But I know what I would trust and don't trust. And the other part, in terms of journal articles, I am brought to the understanding that there's now a group that relooks at journal articles and revokes them, puts out the information that this article is terrible, then the conclusions were wrong. And yet we see in medical practice that those same therapies are being applied, giving the same horrible results. So quality's got to be permeated, in my opinion, and we have to create the incentive for quality as opposed to quantity.
I'd just like to say I agree with Josie. I think that most of the clinical data is pretty good, actually, although the interpretation is very difficult because biology is so complex. We don't have the outcome measures. But it's been published, and this is one of the problems, that say the targets, okay, the published targets, say in cancer. And this, I was told by the directors of the cancer centers, that 70% of those are not replicable. Okay, so you go off and you try to replicate that target, okay, and it's been published in a journal. It's just not on the internet or something. It's been published in a reputable journal, and you can't replicate it to turn it into a drug target, for example. So that's a severe quality problem. I would say the clinical is pretty good. I mean, the quality pharmaceutical investigation is very good because there are all these standards that have to be followed.
So I'm going to say something I think that was implicit in my remarks, and it relates directly to this quality, which is we keep talking about the disincentives to sharing, but many of those disincentives to sharing are incentives for researchers to produce the high-quality primary data. And that's the real dilemma that we have here. And there are many things that have been put forward, and I love both Marty's and John's vision for how the citizen scientists can contribute to the scientific enterprise. But whether that will produce meaningful conclusions, or what has to happen, how that has to be corralled, so it actually can produce reliable conclusions or either pseudo or real trials in the context of this. There is a fantasy that a lot of observational data will equal the quality of trial data, and sometimes that's true, and a lot of times it's not. And we have to be able to tell the difference, and we don't want to detour people into thinking they're engaged in a pseudo-scientific enterprise that doesn't provide the really reliable information. At the same time, we want to harness that energy. So how to do this exactly right? This is a balancing act here on both sides. We don't want to kill the golden goose. We don't want to kill the incentives to produce high-quality data, and we don't want to reduce the incentives to participate in rigorous research, which can, I think, occur to some extent on the internet or in other ways. But this is the challenge here, and it's not just one side or the other.
I just wanted to emphasize, though, that I said I wanted to bootstrap off of patient data. Yeah. Because it's very hard to be able to get pharma companies or cancer centers to give us clinical trial data. In proposing this project, I'm perfectly happy to jumpstart it with a big bolus of data that all of you guys can contribute, because there's a reason to contribute it, because we will use that data together with experts and machine learning in order to be able to compile the best of what we know into what are the actionable subtypes today and how to treat them. And then if the patients get treated and they don't respond as expected, we will find that out very, very quickly and be able to continuously refine those models.
I've seen some of the results of some of that, and I've seen recommendations given to patients who have cancer about what they should be treated with based on their genotype, and some of that was health fraud. Basically, it was so bad, and so I think we have to be very careful with people with a mortal disease, directing them based on a hypothesis. You know, with drug development, it's only about 1 in 10,000 compounds that actually that is a candidate that actually comes through. And even with all the work before clinical work and all the hypotheses and all the testing and in vitro work that's done, only 1 in 20 gets through the process and is actually useful. And that's like with 200 scientists working on it full time. So this is not a trivial exercise. It's not easy. And unitary hypotheses are usually wrong in the area of biology. It's very sad.
Can I ask one question, though, of you, which is that we're not talking about running a trial with putting lots of patients at risk. Imagine that there's one patient who's an advanced patient who has exhausted all other options and is dying, right? And so that patient, with informed consent, has a choice of dying or trying something for which there is a strong community consensus that this might be something worth trying. Okay, this is an opportunity to do, I think, an ethical experiment in a human model much earlier than could be done previously. And likely it won't work. And either way, you'll learn a lot.
Yeah, one of my jobs is that I head the National Center for Complementary and Alternative Medicine, and it won't surprise people in this room to say that I've become a bit of a skeptic about many of these issues. And I do want to echo what Dr. Woodcock has said about the need for caution in this arena. And actually, when you have very large data sets, you can have signals that look significant, even when they really aren't.
Yeah. So I have a colleague who reminds me of that pitfall with large observational data, which is that because you can get such power out of it, you can get precisely the wrong answer.
On the other hand, I think the idea is right, but I think it ought to be done in an I SPY type of manner. We're right now we're talking about taking I SPY to the community. All right, and so there you go to the community doctors, you offer them, we'll genotype your patients, or we'll do these biomarkers on them, and then you offer them therapy, all right, but it isn't in a just sort of like that. It's in okay, you're part of an experiment, okay, that is randomized, okay, and we'll offer you this therapy or that therapy, either of which we think we're in equipoise about whether they could help you or not. And then you adapt as you learn, and you converge on those that are becoming most successful.
Laura Esserman is one of our advisors, and I'd be very open to, the scientists are part of this community, and I think that one of the keys benefits for scientists is when patients are in the same subtype and some respond and some don't, right? That's how science gets done. So I would, I'm not trying to rule out any of those things. I'm not a cowboy. I'm trying very hard to find a way to balance the need for urgency in getting something done with the natural tendency of the world to move at a speed which is frustrating to an internet guy.
Thanks. One of the dangers of data sharing that's come up a couple times this morning is the potential for misleading secondary analyses of that data. And since I consider myself someone who really is for greater openness in data sharing, that worries me tremendously because I can see it as tearing down this huge house that we're trying to build here. And I learn best through case studies and examples. So I'd like to ask for a list of those that have occurred in the past. And one that I've heard an individual mention to me is the Andrew Wakefield case with MMR vaccine. That had nothing to do with a secondary analysis of data and data sharing. So what are the examples that are most poignant? For example, Dr. Rosenblatt, you talked about this danger. I don't know if there are some in your mind, perhaps, that speak to you strongly about this danger.
I have one. The large data sets from the United States Renal Data System failed to identify the dangers associated with high doses of EPO, and in fact suggested that that it was in fact beneficial in the dialysis population.
I would say that observational studies in general have undermined the public's confidence in science, because we have this long history, all right, of fiber will prevent colon cancer, and this will do that, and this will do that, and we announce these things, and the epidemiologists know these are associations, not causally linked. They're presented to the public as causally linked, and then where they debunked, which they all are, then the public says, these scientists don't know anything. They don't know what they're talking about, because these things are announced, and we hear one after the other. Same would be with some of these retrospective subgroup analyses. Fortunately, the public doesn't get so excited about these. So we've had a couple where, okay, this drug causes MI due to some retrospective meta-analysis, and then, you know, you do a randomized study, and lo and behold, you don't see it, okay? Well, nobody's going to get excited one way or other about that, because they didn't know very much about it. But there are quite a few of these. And the OMOP collaboration that is going on and testing methods for observational studies, okay, has shown that the ability of these studies to predict small effects is very, very poor and can be wrong either way. So the same most of these subgroup analyses will probably be meta-analysis. So you combine various sources of data and do a meta-analysis, and they are subject also to various biases.
I think I'm all in favor of these secondary analyses, but they don't prevent us from needing the randomized, rigorous.
Oh yeah, we just thought about the best one, the one that caused the most feeling of betrayal, okay, amongst the public was estrogen and preventing myocardial infarction, okay. I was told years ago, I was told FDA should approve this drug for prevention of MI, because they had calculated how many lives would be saved, okay, by approving estrogen for prevention of MI. And FDA, of course, refused, because there had been no randomized studies on this, and the rest is history.
And some of us who've run these trials end up being criticized because the observational data has already given us an answer, and we're told our trials are unethical, and then the results are surprising.
So what I've learned from this evening, that I didn't realize before, is that data can be bad and analyses can be invalid. And this is raised by data sharing, that observational data needs to be confirmed by randomized trial. This has got nothing to do with data sharing whatsoever, okay? Something like the WHI and the whole epidemiology that was based on has nothing to do with data sharing. It worked, the process worked exactly how it should have worked. I never believed those results. Okay, this 15% decrease in heart disease I thought was ludicrous to say that we could measure that in the observational studies. And therefore I supported the randomized trials. I thought the FDA did exactly the right thing. I don't think shutting off it. What if we'd said the Nurses' Health Study, let's restrict those data, let's not share them, so that
saying we shouldn't share data.
Well, hang on, but that was the point, is that people are bringing up as an argument against
No,
data
I
sharing.
actually don't think that's at all. I think that
that's
people are
explicitly
being,
been raised by the panel.
Not
is
at
that
all.
it is a well, people are nodding with me here.
No, we're just talking about
not only that,
potential limitations that people who work in the field should appreciate, just like there's potential limitations with the data when it's generated in its original form. So we're just go into fields with your eyes open. Nobody's saying not to use it. I think that's overreacting to what you've heard, in all honesty.
Well, let me say, it's not overreacting, because I'm a statistician, and I have, if you go back and read my article, the very first thing that was basically said in response to my request for data was, I'm not going to give you your data because you might do an invalid analysis of it, okay? So this is not an overreaction. This is widely used to prevent individuals accessing data. And
people who do original research get bad reviews, get criticism.
I know.
That's part of the scientific discourse.
No.
And that's honestly, I think that's what's going on here. Nobody's saying. I think everybody, including myself, came in here advocating for data sharing.
Okay.
Just to be on the record.
All right.
And clear about it.
Well,
okay.
great. Well, then let's be very clear about this. You advocate data sharing. You're raising this as a potential problem. You have to be very.
Talked about the benefits.
Right. Okay. What I'm saying is, this, as a practicing statistician who has tried to obtain data sets, and people have said, you can't have them in case invalid analyses are done.
Yeah.
I think we have to be very, very careful when we have people at the panel saying, well, this is a downside of data sharing. So
And I've been a practicing physician, and we have to be very careful about the best interests of patients. And we're trying to find a way here, all of us together, to reconcile that.
The concerns I meant to echo were concerns about internet-based analyses. For example, data on the predictability of a given SNP of disease. And those are areas where I think a number of us are concerned that very large data sets might be generated without proper attention to the true predictive value of that data. And that's really being driven not by scientific reanalysis of data, but by a marketplace that's using data in ways that may not be appropriate.
So I just want to echo something that Marty Tenenbaum pointed out, and the dialogue with Janet Woodcock. With the I-SPY model, with the foundation of the NIH, a number of us have been involved in another experiment out there called the Alzheimer's Disease Neuroimaging Initiative, which is, in fact, sort of what I think you want. It's naturalistic. Society shares. Just to remind everybody, we didn't put it on the program, but just to remind people, I mean, because it is the example that we took the risk. We started planning this in 2000. All the data goes on the internet immediately. Genetic data. Somehow, I don't know how we got around it. We got the privacy issues. I mean, we have people's genetic data. We have their brain imaging data. We have their cognitive data, healthy volunteers, MCI. This has been repeated. It's going to go on for another several years. As far as I know, we're sharing. On the data citations, I've suddenly found that my CV is much longer than I ever dreamed it would be, because I get, just following the model that Dr. Acton puts out, we have this whole long list of us. We all get cited whenever a paper shows up. I think, wow. So all this is going on. And so far, I'm not aware of any downsides, okay? That's all I'm trying to say.
Well, this is
So,
these observational.
I just wanted to point that out.
Yeah, these
You can do
observational
this.
natural history studies. Academies has stimulated drug development in Alzheimer's, which is a national priority, and so it has also enabled it because we understand the biomarkers to look for, and we understand more about the course of the disease. So that's been a tremendous enabler. And basically, what they did is follow people and got biomarkers on them and saw what happened to them. Yeah, but they did it in a rigorous manner, right?
The one comment that I made in passing was that cancer being hundreds or thousands of diseases, hundreds of targeted therapies having to be used in cocktails maybe to manage it like AIDS. I don't know how we can continue the current system of trials. It'll break down. There's not enough patients to go
Current
around.
system of trials is completely broken already, right? And so, it could break down further. We need to have wide community-based intake and match people to investigational therapies based on their signatures. Okay, agreed. But I'm saying we need to do that in a organized, systematic manner and do that from the get-go. I think just sort of collecting, because collecting people, we don't know when their tumor was biopsied, we don't know what test was applied to their tumor. There's enough complexity already without adding this other layer of uncertainty on it.
I would love to work with you, the FDA, and others in this room to make that happen. I mean, that is, my own organization, Cancer Commons, is meant to be a connecting point, not to do any work per se, but to connect I-SPY to thousands of patients through advocacy groups, for example.
Yeah, well, we're talking about a large lung cancer trial that's an ongoing trial with the Friends of Cancer Research soon that would, take in agents continuously. So the trial is constantly stood up and is a resource, and the patients and the investigational agents are taken into that with the actual goal of matching the profiles against the drugs.
So I would love to figure out a way to connect to that, given that we have a very active lung cancer rapid learning community that's going right now.
One last question quickly.
It goes back to the elementary stuff we all learned undergraduate. Observational data leads to hypothesis, and then you test the hypothesis. You don't act only on the observational data.
Thanks. So I'm going to conclude our 2 days and ask you to thank our last panel for wrapping us up with some vigor.